Hello, my name is Simona Jasaityte, and for the last couple of years, I've been coordinating the study called the Parkinson's and Movement Disorders Families Project, or PFP in short. And here with me, I have my colleague Matilda, and I'll let her introduce herself briefly.
Hi, so yeah, just a quick introduction from me. My name is Matilda, as Simona said. I've just come on board the PFP study, and so I'm in the process of taking over from Simona as the new study coordinator. I'm really looking forward to learning more about the study and getting more and more involved and see where the study and the research takes us. So I'll hand back over to Simona to talk more about the objectives and what has been achieved so far.
Okay, thank you. Okay, so I'll take over for the presentation, and I'll just start with a very brief background to the study. So Parkinson's is thought to have multiple causes. It is thought to involve causative genes and susceptibility genetic variants that are modulated by environmental exposures, and family history, as well as early onset of Parkinson's disease are thought to be indicators of potential genetic involvement in the causing of disease. And however, for the vast majority of these patients in these groups, so familial patients, those who have other family members who are affected, and individuals with very early disease onset, a genetic cause has not been identified yet. And this in turn leads us to hypothesize that there are more gene mutations that are yet to be discovered. And so the main objective of this study in particular is to find these new gene mutations that we have not identified yet. Mutations that either predispose to developing Parkinson's disease and other movement disorders or directly cause it. And we're hoping that by learning more about the biology of this disease, we are going to be informing better and improved tests and treatments in the future. So who is eligible to take part in the study? So the first person who takes part in the study from the family is referred to as the index case in this research. So an index case is someone who has a clinical diagnosis of Parkinson's disease. And in addition to that, they either are an early onset patient, so they develop their symptoms at or before the age of 45, or they are a familial patient. So they have family history of Parkinson's disease, at least one other family member who is affected by Parkinson's or another movement disorder. So those are kind of the main two patient groups. And in addition to that, we're very much interested in recruiting family members of those who are affected. So we are recruiting both affected and unaffected family members. And because as relatives, we share a lot of DNA, and it is very helpful to compare the DNA of those who are affected and those who are unaffected, so then we can see the difference and see which are the bits that are different, and we can analyze them as potentially being those that are causing or contributing to the disease. So the study involves providing a blood sample, which is later used for genetic analysis, and also completing a couple of questionnaires, which takes about an hour. Question is about your family history, about your medical history, about the symptoms you experience, movement and non-movement related. And then also completing an in-person assessment in one of our 43 sites across the UK. And so the in-person assessment involves a movement assessment as well as a very brief cognitive assessment. In terms of sample analysis, so we have used multiple different types of analyses to analyze the samples. Currently, we are focused on whole genome sequencing all of the samples in the study, and we have done so for the majority, and we are waiting for data to return so we could analyze it. And whole genome sequencing is really the analysis that shows us the whole genome. It allows us to work on identifying these new genetic mutations that we are kind of not aware of yet. And we're completing this analysis with the help of Global Parkinson's Genetics Program, which is an international project. So they're kind of completing the data analysis, the sample analysis for us, and returning data back to us, and we're waiting for it right now. And we have recently written a baseline paper, which is essentially an overview of the study so far, what kind of clinical data has been collected, and the mutations that we have identified in our patients so far. And we are hoping to publish this paper soon, hopefully. And in terms of the results, so we currently have recruited over 1,600 participants in the study so far. So 1,300 of the participants are the index cases that I mentioned before. We have 75 affected relatives and 252 unaffected relatives. And I mentioned the preliminary analysis that we have completed so far. And so that was done with 712 families who took part. And so we found that 51 individuals in these families who were affected, they carried a pathogenic variant, so a mutation that causes Parkinson's disease. So that is 7.1% of individuals in our cohort carried a pathogenic mutation causing Parkinson's. And LRRK2, which is an autosomal dominant mutation, was the most common, and this is supported by previous research. It usually is the most common causative mutation in Parkinson's. So this mutation was found in 4% of our index cases, and biallelic mutations in a recessive gene called Parkin or Park2 were the second most frequently found mutation, and it explained Parkinson's in 2.4% of families. So as you can see, only the absolute minority of our patients were found to carry a causative mutation in Parkinson's, only 7.1%. And this leads us to believe that there are likely to be found further monogenic causes, because all these patients, they have family history, they have very early onset of disease, indicating that potentially we couldn't find a mutation in these patients yet because we have not identified this mutation so far. So further analysis is on the way, and yeah, we're hoping to bring more exciting results to you in the future. Thank you very much for your time.